In this video, we'll be looking at questions 6 through 10 of the Set 1 Genetics in the Topical Review Book Company's Living Environment Core Curriculum Workbook, 2nd edition. Question 6. Which statement best describes human insulin that is produced by genetically engineered bacteria? So this question is really almost exactly referred to in diagram 13 on page 35. To review that diagram, a restriction enzyme is used to cut open the DNA plasmid of the bacterium, and another restriction enzyme is used to cut with the same base sequence targeted the human insulin gene. That human insulin gene is then inserted into the bacterial plasmid. Now, the bacterium will produce the human insulin hormone. And every time that bacterium divides, the offspring that are genetically identical to the parent bacterium will also produce the insulin hormone. This allows for human insulin hormone to be produced in an efficient and inexpensive way. So, to read the options. Option 1. The insulin will not function normally in humans because it's produced by a bacteria. While it's true that the insulin is produced by a bacterium, it's still the human protein. The mechanism used by the bacterium to produce proteins is the same mechanism used in humans to produce proteins. So the code for a human protein, even in a bacterium, will still produce the human protein. So option 1 is incorrect. Option 2, this insulin is produced as a result of human insulin being inserted to bacterial cells. It's not the insulin that's being inserted into cells, it's the DNA code that makes the insulin hormone protein. So option 2 is incorrect. Option 3, the insulin is produced as a result of exposing bacterial cells to radiation, which produces a mutation. Option 3 is incorrect because genetic engineering is not hoping that a mutation occurs and does what you want. Genetic engineering is a human going in and changing the DNA specifically and on purpose. Option 4. This insulin may have fewer side effects than the insulin previously extracted from the pancreas of other animals. So before we had genetic engineering technology and we could do something like this, a lot of times we were using pig insulin for diabetics. This was expensive, and since it wasn't human insulin, it was not entirely compatible with our human bodies. Now that we can produce human insulin from bacteria quickly, efficiently, and cost-effective, we're seeing fewer side effects because the insulin is normal human insulin. So the answer to question 4, or question 6, is option 4. Question 7. Individual cells can be isolated from a mature plant and grown with special mixtures of growth hormones to produce a number of genetically identical plants. So as I'm reading the question, I'm seeing cells can be isolated, which means removed, and then they're used to make genetically identical plants. Those are the important words that I'm focusing on as I move into reading the options. To finish the question, this process is known as option 1, cloning. I know that cloning produces identical offspring, but again, I'm going to look at all of the options because there may be a better option in the list. Option 2, meiotic cell division. I know that creates gametes that have half of the DNA of a normal cell, so that's not going to be my option. Option 3, recombinant DNA technology. Recombining DNA will create a unique individual and not an identical copy, so that is going to be incorrect. Selective breeding, again, that's when humans select desirable traits from individuals and have them reproduce to produce a genetically unique offspring that has favorable traits. So now I know that number 4 is not my answer. So numbers 2, 3, and 4 are all creating genetically unique individuals, while cloning is creating an identical individual from the parent. So my answer is number 1. Question 8. Which statement best describes the relationship between the terms chromosomes, genes, and nuclei? From here, I want to go back to the overview from the reading, and we see a smallest to largest setup here. So starting with the smallest, we have genes, which are segments of DNA. DNA is stored in major structures called chromosomes. Those chromosomes are found in the nucleus of a cell. So looking at the options, we're looking for the relationship between chromosomes, genes, and nuclei, which is the plural of nucleus. Option 1, chromosomes are found on genes. That's incorrect because genes are parts of DNA which are bundled into chromosomes. So genes are found on chromosomes. Option number 1 is incorrect. Chromosomes are found in nuclei. That is correct, right? So the chromosome is smaller and is found in the nucleus. Nuclei are found in genes. That is incorrect. Genes are incredibly small, just sections of DNA, which is stored in the nucleus. Option number 3, genes are found on chromosomes. That is correct because genes are segments of DNA. DNA is stored in chromosomes, so the genes would be stored in chromosomes. Chromosomes are found in nuclei. That is also correct. So chromosomes are stored in the nucleus of a cell for safekeeping, and that's going to be our answer. Question 9. The diagram below represents a section of a molecule that carries genetic information. So that molecule that carries genetic information is DNA. We see the double helix structure of the DNA in the diagram with 2 bands winding around each other. The pattern of numbers represents. So if we look close, we have the phosphate and sugar backbones, and then numbers sort of making the rungs of this ladder if it was a ladder. Those numbers represent 1, the sequence of paired bases. In DNA, we have the sugar and phosphate backbone, and then the molecular bases that would form the rung of the double helix ladder. So 1 and 2 are going to pair together, and 4 and 3 are going to pair together. So 1 is going to be our correct answer. The numbers are the paired bases of a DNA molecule. And just as a refresher, those bases are adenine, A, which pairs with thymine, T, and cytosine, C, which pairs with guanine, G. Looking at the other options. 2, the order of proteins in a gene. So it's the order or the sequence of paired bases that will determine the sequence of amino acids in a protein. So the proteins are not found on a gene, but genes code for proteins. 3, the folds of an amino acid. Amino acids are not folded. However, extremely long chains of amino acids coded for by genes in DNA will fold over each other and form a protein. So number 3 is incorrect as well. The positions of gene mutations. Since we don't know what the original sequence was in our molecule up here, we don't know if there are any mutations. Without knowing the original sequence, we cannot determine if there's a mutation in the current sequence. So 4 is incorrect. Number 10. Asexually reproducing organisms pass on hereditary information as. So again, before we read the options, we want to analyze the question. First, asexually reproducing organisms reproduce with the process of mitosis, and the hereditary information is DNA. So reading through the options. 1, sequences of A, T, C, and G. They're talking about the molecular bases, adenine, thymine, cytosine, and guanine. Let's move on and see if the other options are better, but that's a good one. 2. Chains of complex amino acids. Amino acids are not complex. When you chain them together, that chain becomes complex, but the amino acids themselves are not complex. Incorrect. 3. Folded protein molecules. Proteins do not store hereditary information. That belongs to the nucleic acids like DNA and RNA. 4. Simple inorganic sugars. Inorganic means they don't contain carbon and hydrogen. Sugars, carbohydrates, all contain carbon and hydrogen, so all sugars are organic. Also, they don't pass on hereditary information. So, the hereditary information, the traits that we have, are coded for in our genes, which are sequences or orders of adenine, thymine, cytosine, and guanine, those molecular bases. All of those sequences determine which amino acids are taken next, and that will determine the shape of the protein and ultimately the function of the protein. So the answer here is option 1. Asexually reproducing organisms pass on hereditary information as sequences of adenine, thymine, cytosine, and guanine, ATCG. Also, this is true for sexually reproducing organisms. All organisms on Earth pass on hereditary information to their offspring through DNA with sequences of A, T, C, and G. And this will conclude the second video of questions 6 through 10 for Set 1 genetics questions from the Topical Review Book Company's Living Environment Core Curriculum Workbook, 2nd edition. Look out for the next video, which will cover questions 11 through 15.